Hi everyone, this is our lecture on the pentose phosphate pathway. So let's get started. To get a clinical perspective, let's look at a quick flash case. Let's say a 20-year-old African-American male presents to the clinic because his college roommate noticed his eyes appeared yellow. When asked if he had been prescribed any new medications or had any recent travel, he responded, "The only kind of travel I've done recently is try the new Mediterranean restaurant next to campus last weekend." Peripheral blood smear shows cells as shown in the image. Let's keep this in mind as we go through the lecture. Our learning objectives are to recognize the role of the pentose phosphate pathway in metabolism, and to recall the sites where the pentose phosphate pathway occurs. So first, let's get oriented. Here is our summary of pathways and how they are all interconnected. And the pentose phosphate pathway is right here. And it's also called the HMP or hexose monophosphate shunt. Let's get a closer look. Now that we're closer, we can see our friend glucose goes through glycolysis and turns into glucose 6-phosphate and then fructose 6-phosphate, which is great. But in certain sites, glucose 6-phosphate can alternatively take another path into the HMP shunt and become 6-phosphogluconolactone and then ribulose 5-phosphate to again turn into fructose 6-phosphate. Why would the body go into the trouble of having an entirely different pathway to get to the same thing? Have you ever studied for an exam with a classmate and you had your own method of studying, maybe by watching videos, while your classmate didn't watch any videos at all, but instead just used flashcards? And did you ever think maybe their method was better, so you tried it? Maybe there was something to gain by taking a different path. Or maybe this path is useful for certain individuals, or certain sites in the body, or certain circumstances. Let's take a closer look. As a disclaimer, the previous slide was a big overview and it has a different intermediate listed, so don't get too obsessed with that. Focus on the high yield points here. So in the pentose phosphate pathway, glucose 6-phosphate is converted to 6-phosphogluconate by the rate-limiting enzyme glucose 6-phosphate dehydrogenase. And in that process, an NADPH is formed. Well, what's so great about NADPH? Remember, it's one of our universal electron acceptors and is used in anabolic processes like steroid and fatty acid synthesis as a supply of reducing equivalents. We need it for reductive reactions, like needing glutathione reduction in red blood cells. As we keep going along the shunt, 6-phosphogluconate is converted to ribulose 5-phosphate, and in the process, it makes another NADPH and carbon dioxide as well. Now, this phase of the pentose phosphate pathway is known as the oxidative phase, which is irreversible and occurs in the cytoplasm. The next phase is the non-oxidative phase, which is reversible and also occurs in the cytoplasm. Now, ribulose 5-phosphate is converted to ribose 5-phosphate, which is great, but what do we need ribose for? You're right if you're thinking of nucleotide synthesis. As for the rest, as shown here, it completes the shunt and can help us return to glycolysis and gluconeogenesis. Okay, let's do a recap. The pentose phosphate pathway is also called the HMP shunt, and it provides a source of NADPH from abundantly available glucose 6-phosphate. Why do we need NADPH again? Why would we go through the trouble of another pathway or a shunt to make it? Well, we need it for reductive reactions like glutathione reduction inside red blood cells, like in this diagram, and in anabolic processes like fatty acid and cholesterol biosynthesis. Well, this makes sense, right? Can you remember what the rate-determining enzyme of cholesterol synthesis is? You're right if you're thinking, it's HMG-CoA reductase. Remember, the pentose phosphate pathway also yields ribose for nucleotide synthesis. Remember, there are two distinct phases that occur both in the cytoplasm. Can you remember what they're called? Yes, they're the oxidative and non-oxidative phases. What's great about this shunt is that it gives us NADPH and ribose, but no ATP is used or produced. Where then would you expect the pentose phosphate pathway to come into play? Where do we need to synthesize and reduce things? So you're right if you're thinking its sites are in the lactating mammary glands, the liver, the adrenal cortex, which is where fatty acid and steroid synthesis occur, and in the red blood cells. Okay, that was a lot of information. Let's do a quick quiz. Can you remember what the products are of the pentose phosphate pathway? Great if you said NADPH, carbon dioxide, and ribose 5-phosphate. Well, let's get back to our case. So we have a 20-year-old with yellow eyes or jaundice after eating Mediterranean food for the first time. What can you see in the image? Yep, this is a degmacyte, or a bite cell, which is a result of phagocytic removal of Heinz bodies by splenic macrophages in glucose 6-phosphate dehydrogenase deficiency, the rate-determining enzyme of the pentose phosphate pathway. With G6PD deficiency, we won't make enough NADPH to keep glutathione reduced. So this can lead to a hemolytic anemia, since red blood cells won't defend well against oxidizing agents. Now, our typically oxidizing agents can include antimalarials or anti-tuberculosis drugs, but also fava beans, which are common in Mediterranean food. So, what's the bottom line? The pentose phosphate pathway, or H&P shunt, provides a source of NADPH from abundantly available glucose 6-phosphate, which is required for reductive reactions. It has two distinct phases, oxidative and non-oxidative phase, which both occur in the cytoplasm. And the sites of the pentose phosphate pathway are in the lactating mammary glands, the liver, the adrenal cortex, and the red blood cells. That's all I have for you here. Please give this video a thumbs up if you liked it, and it'll give you the opportunity to submit a comment if you have any feedback or questions. Thanks for joining me. Study hard.